A very good morning to one and all. I am Dr. Wasim, Senior Resident in Department of Ophthalmology, Sri Lakshmi Narayana Institute of Medical Sciences, Pondicherry. Today, we will be talking about the physiology of the cornea. From the previous classes, we have understood the anatomy of the eye, the anatomy of the cornea. Hope you all remember the anatomy of the cornea. The cornea contains of 5 layers, 6 including the Duas layer, starting from the epithelial layer, the Bowman's membrane, the stroma, the Decimate membrane, including the predecimates membrane or the Duas layer, the Decimate membrane, and finally the endothelial layer. Today, let us look into the physiology of the cornea, like how the transparency of the cornea is maintained and how the metabolism or the transport of nutrients and ions takes place within the cornea. And to an extent, we will also understand the pharmacology basis of cornea and how drugs penetrate into the cornea. Let us go into the class today. Okay, so starting with the functions of a cornea. Okay, so the cornea, as we all know, is the anterior 1/6 part of the fibrous coat of the eye. It is a clear, transparent layer of tissue. Apart from the protective function and the refractive function, it has a very important role in the refractive media of the eye. As we already discussed in the sections on anatomy of the cornea, we said that the cornea contributes about 3/4 of the refraction of the eye. The remaining 1/4 is only contributed by the lens. So 3/4 of the refraction is contributed by the cornea. So the cornea or the air tear film corneal interface is the major refractive surface of the eye. So it is the most important refractive media in the eye. Okay, we already know that the transparency of cornea results from the uniform spacing of collagen fibrils in the substantia propria. So what happens is, if any increase in the tissue fluid between these fibrils, or when the distance between the fibrils gets separated, or the uniform architecture of these fibrils is lost, then we get cloudiness or the edema in the cornea. As we all know that comparing the cornea to the sclera, in the sclera, these fibrils are irregularly arranged. That is why we know that the sclera is opaque. But unlike in the cornea, where these fibrils are uniformly arranged, and the thickness of the fibrils, as we already discussed in the class on anatomy, it is 25 to 35 microns, and the interfibrillar distance is also about 40 microns, which is half the wavelength of light. This regular arrangement, as we all discussed, causes a destructive interference, thereby allowing the light to pass through the cornea. Okay, so when this architecture gets distorted, or when this architecture is lost, what we get is a cloudiness of the cornea, or a corneal edema to say. Okay, so the endothelium and the epithelium also play a major role in limiting the fluid uptake by the corneal stroma. So the epithelium and the endothelium are in a constant process of maintaining the transparency, thereby maintaining the amount of fluid, amount of water that is present within the corneal stroma. Let us consider what is the composition of the cornea. We all know that the cornea is mostly made up of water. Water content of the cornea is about 78%. The remaining dry weight is the collagen. Okay? So the collagen contributes about 15% of the total weight of the cornea, or the major source of the dry weight. In this 15%, the type 1 collagens are the most, contributing to 50 to 55% of the entire dry weight, followed by the type 6 collagen, which is 25 to 30%, and the type 4 collagen, which is 8 to 10%. Minimal amount, or less than 1%, is contributed by the type 3 collagens. Apart from collagens, there are also other proteins which are present in the cornea, like the other proteins which contribute about 5% of the total weight of the cornea. Okay, these other proteins are especially keratin sulfate, which contributes 0.7%, chondroitin or dermaton sulfate, which contributes 0.3% of the corneal weight, and hyaluronic acid and other salts, which contribute about 1%. So now coming to the biochemical composition of the cornea. Okay, considering each individual layer, we will consider each individual layer separately. First, to start with the epithelium. The epithelium forms 10% of the total weight of the cornea. As we all know that the epithelium, it is a stratified squamous non-keratinized epithelium which contains of a basement membrane, a basal cell layer, a wing cell layer, and a superficial layer, as we all discussed in the class on anatomy. Now, this epithelium contributes 10% of the total weight of the cornea. The water content of the epithelium is about 70% of the wet weight we all discussed, and the protein synthesis in the epithelium is the highest because there is a constant turnover of the epithelial cells. Like as we all discussed in anatomy, the epithelial cells from the limbal cells are pushed towards the center, thereby contributing to the basal cells. From the basal cells, they gradually move up. When they move up, they lose their architecture and they become more flattened. We all seen in the class on anatomy that they become wing cells, and the wing cells further flatten up and become the superficial cells. These superficial cells are periodically shed into the tear film. Okay, so the protein synthesis or the protein turnover in the epithelium is the highest. Apart from this, lipids do contribute a significant amount in the architecture of the epithelium, and the major lipids are the phospholipids and the cholesterol. Okay, so these epithelium is also contain enzymes which are necessary for the Krebs cycle and the glycolysis. Okay. Apart from this, the other proteins or other nutrients which are present in the epithelium are acetylcholine, adenosine triphosphate, glycogen, and glutathione. The major electrolytes in the epithelial layer are sodium, potassium, and chloride ions. Now, coming to the stroma contains 80% water and 20% solid. We already discussed. The 20% solid are mostly the collagens. Okay? The collagens, as we know, it's a type 1, type 5, type 12, type 11, and type 10 and type 4. The soluble proteins. Among the soluble proteins are the albumins, immunoglobulins, and the glycoproteins. As we all discussed in anatomy, the proteoglycans, that is, the glycosaminoglycan fractions contribute the major uh, proteins in the stroma. The glycosaminoglycans include the keratin sulfate, which contributes 50% of the total weight of the stroma, followed by chondroitin sulfate and chondroitin itself contributing 25% each to the dry weight of the stroma. Okay. Apart from this, there are glycolytic and Krebs cycle enzymes. Okay, and there are also matrix metalloproteinases. Like matrix metalloproteinase 1 or the collagenase, matrix metalloprotein 2 or the gelatinase A, and the matrix metalloprotein 3 or the stromalysine 1. Okay, the stroma also contains electrolytes and salts. Now coming to the Decimate membrane. The Decimate membrane, as you know, is constantly laid down by the endothelium. Unlike the Bowman's membrane, which we have already discussed, the Decimate membrane is the true basement membrane of the endothelial cells. The Decimate membrane is composed of collagen to an extent of 73% and glycoproteins. The collagen of the Decimate membrane is insoluble. It's very important, and it is extremely resistant to chemical and enzymatic action. As we already told that in case of bacterial ulcers and in case of fungal ulcers, even when the entire epithelium stroma is eaten away, because the Decimate membrane is tough, these bacteria or the fungal or the viral toxins do not destroy the Decimate membrane. That way the Decimate membrane is held intact, and in case when there is an increase in intraocular pressure, this Decimate membrane bulges out through the defect. That's what we call it as a decimatocele. Okay, so the Decimate membrane, the collagen of the Decimate membrane is insoluble and is extremely resistant to chemical and enzymatic action. Coming to the last layer, the endothelium. As we all know that the endothelium is a single layer structure, hexagonal arrangement in a mosaic pattern, and we know that the endothelial cells do not regenerate, And what happens is when an endothelial cell is lost with aging, the neighboring endothelial cells enlarge and occupy the space, which we call it as polymegatism. And when they change their shape, we call it as pleomorphism. These all we have discussed in anatomy. Okay, so the endothelium is a single layer structure. It contains the enzymes for glycolysis and Krebs cycle. Okay. Now coming to the corneal metabolism. Okay. So as we already said that the most active layer of metabolism in the cornea is the epithelium and the endothelium. Okay. So the energy is in the form of adenosine triphosphate, which is generated by the breakdown of glucose. The most important source of glucose is of course the aqueous humor, partly by the perilimbal vessels and a little amount from the tear film. Okay, so the source of nutrients for the corneal metabolism and the metabolic pathway are the oxygen, like from the air, the glucose, and the amino acids. Okay, coming to the each nutrient individually. Coming to oxygen, oxygen for the epithelium, it's derived from the tear film and the limbal capillaries. As we have discussed in anatomy itself, the blood supply of the cornea, there is a perilimbal arcade, and the perilimbal vessels penetrate into the corneal periphery for a distance of about 1 millimeter. So it is from these perilimbal vessels the oxygen diffuses. Also, from the tear film directly, the oxygen also diffuses. We will read in detail when we read the pathological features of cornea, like in case of contact lens wearers, and in case of other corneal disorders, when there is a decreased supply of oxygen to the cornea becomes cloudy. Okay. So the oxygen required by the epithelium is 1 by 10th of that available from the atmosphere when the eyes are open, and it's about 1/4 available from the palpebral conjunctiva when the eyes are closed. Okay. So the endothelium derives its oxygen from the aqueous humor, and the total corneal oxygen consumption is 9.5 ml of O2 per centimeter square per hour. Okay. Next, coming to the next major metabolite, the glucose. Glucose is a primary metabolic substrate for the epithelial cells, the stromal keratocytes, and the endothelium. So, glucose is transported again from the aqueous humor through diffusion from the perilimbal vessels, and glucose from the tear film also contributes a very minimal or a meager amount for the metabolism of the cornea. So in case of an exogenous supply of glucose, there are also glycogen stored in the corneal epithelium. The cornea also contain glycogen stored like the liver and other cells and tissues in the body. So this glycogen is also broken down into glucose and utilized for the production of energy in the cornea like other tissues. Okay, so the rate of glucose consumption of the cornea is 100 milligrams of glucose is consumed per centimeter square per hour, and the 90%, that is the major chunk of glucose, is consumed by the epithelial layers because of the constant turnover of epithelium. We all said that the epithelium keeps shedding every day, and the basal cells move up, forming the wing cell superficial layers of the epithelium. Okay, so coming to the glucose metabolism. So glucose is metabolized by the cornea by three pathways. There is an anaerobic glycolysis, that is what happens in anaerobic glycolysis, that is in the absence of oxygen, the glucose is converted into lactic acid, and there is a production of 2 adenosine triphosphate. Okay, this is the anaerobic, like in the absence of oxygen. This is all we know from our biochemistry. Okay, the next major metabolic pathway is the Krebs tricarboxylic acid pathway, or the Krebs cycle. In this Krebs cycle, the end product of glucose metabolism is carbon dioxide and water, with a major generation of almost 36 molecules of adenosine triphosphate. And this is the major source of, this is the highest source of energy for the cornea. Apart from this, the third pathway is the hexose monophosphate shunt pathway. This hexose monophosphate shunt pathway is mostly not used for energy production. It is used for generation of nucleic acids like NADPH or ribose, and NADPH produced in this process, in this hexose monophosphate chain, is used for utilized in lipid synthesis and synthesis of various other intracellular molecules by the cornea. The ribose, which is produced as a byproduct of this hexose monophosphate chain, is also used to build DNAs, RNAs, and other nucleic acids. So these are the major three pathways of corneal metabolism of glucose. Now, coming to the glucose metabolism in detail, of the entire amount of glucose that is available to the cornea, only 12% of the glucose is metabolized through the tricarboxylic acid cycle. That is, only 12% is metabolized, and there is a generation of 36 molecules of adenosine triphosphate. Okay, so the major metabolic pathway of the cornea is the anaerobic cycle. So in the anaerobic cycle, as we all said, that there is only generation of two molecules of adenosine triphosphate along with the generation of lactic acid as an end product. So with the generation of lactic acid, there is an accumulation of lactate in case of even in case of aerobic metabolism. So even under the case of aerobic metabolism, there is also production of lactic acid. What happens to this lactic acid is this lactic acid is eliminated from the cornea by a process of diffusion through the epithelium. So even in case of aerobic conditions, mostly the cornea prefers to undergo production of lactic acid and two molecules of ATP. Krebs cycles contributes only 12% of the total energy source of the cornea. Okay. Apart from this, in the epithelium and the endothelium, the hexose monophosphate pathway breaks down 35% to 65% of the glucose, but the stroma metabolizes very little via this pathway. The hexose monophosphate shunt is a major glucose pathway for the epithelium and endothelium because of their active turnover of cells or their active metabolism, while the stroma mostly it is used for its transparency or detergents, and it does not prefer the hexose monophosphate pathway. Okay, so pyruvic acid is a end product of glucose in case of tricarboxylic acid pathway. Apart from glucose, amino acids are also supplied to the cornea. They are mostly supplied from the aqueous humor by passive diffusion. The requirement of amino acid for the synthesis is 10 milligrams per hour of protein. So 10 milligrams per hour of protein for constant shedding and replacement of the epithelial cells of the cornea. Okay, now let us look at the lamellar arrangement of the cornea. That is, what contributes to the corneal transparency is the regular arrangement of the collagen. We already saw in the class of anatomy or the discussion of anatomy that unlike sclera, the corneal fibers are arranged in lamellae, that is in regular layers, and the keratocytes are interspersed in this lamellae, and this lamellae have a uniform, the collagen of this lamellae have a uniform diameter of 25 to 35 microns, and the interfibrillary diameter is also constant at a range of 41 microns, which causes destructive interference. So two theories were proposed for this lamellar arrangement. One was the Morris theory, which is the most commonly accepted, which was proposed in 1957, and the other was the Goldman and Benedict's theory, which was proposed in 1967. Okay. According to the Morris theory, the stromal collagen fibrils are of equal diameter, that is what we said, 275 to 350 angstrom units, and they are equal distance from each other, and they are arranged as a lattice with interfibrillary spacing that is less than that of the wavelength of light. It is smaller than that. It is even half the wavelength of light. So when the spacing between the fibrils is half the wavelength of light, there is no any interference. That is no any destructive. There is a destructive interference. Thereby, the light is allowed to pass through the corneas. Okay. Apart from this, Morris, he explains that because of the smaller diameter and regularity of the separation, the backscattered light would be almost completely suppressed by the destructive interference. That is, reflection is almost completely suppressed Thereby, there is only refraction of the light, and the light passes through the cornea into the eye to fall on the retina. Okay. The next theory was Goldman and Benedict's theory. They suggested that there is a perfect crystalline lattice periodicity. The perfect crystalline lattice periodicity is not always necessary for sufficient destructive interference. They explained that if the fibrillar separation and the diameter is less than a third of the wavelength of incident light, almost perfect transparency will be there. That is, they said that regular arrangement is not at all necessary, but if the separation and the diameter is less than one third of wavelength, then almost the cornea will be transparent. The stromal fibrillations are small in relation to the light, and they do not interfere with light transmission unless they are half than one half of the wavelength of light. That is, if the size of the fibrils, or if the interfibrillary distance is less than half of wavelength, then there is no any opacity, or there is no any backscatter, or there is no any reflection of the light. Rather, the light is allowed to pass through the cornea, thereby causing refraction in the cornea. Okay, but the major demerits or the defects in these two theories is both the theories fail to explain why there is a corneal clouding which occurs during raised IOP, and there is a corneal clearing occurs on reduction of IOP. That is, when there is an increase in intraocular pressure, why is there a clouding happening? And when the intraocular pressure is reduced, and why is that the cornea regains its original transparency? This fact was not clearly explained by both the Morris theory and the Goldman-Benedict's theory. Okay, now let us see how the corneal transparency is maintained. The corneal transparency is maintained by various physiological factors. That is, the stromal swelling pressure. That is, you can see in this diagram, there is a stromal swelling pressure which allows the stroma to swell or increase in size. And there are also metabolic pumps. There are also metabolic pumps both in the epithelium and the endothelium, which constantly move water from inside the cornea to the aqueous humor by the endothelium and towards the tear film in case of the epithelium. And also, there are also barrier functions, the epithelial barrier and the endothelial barrier, which do not allow water to enter into the cornea, thereby maintaining the detergent state of the cornea, that is a partly clear state of the cornea. Okay. Apart from this, there is a constant evaporation of tear film from the surface of the cornea. This evaporation of the tear film draws water from the epithelium, which in turn draws water from the stroma, thereby causing the cornea to remain in a partly dehydrated state. Okay, the rest also, there is also contribution by the intraocular pressure. The intraocular pressure constantly exerts a pressure on the endothelium, thereby pushing the water, trying to push the water into the cornea in case of an increased intraocular pressure, or allowing the water to come back into the aqueous humor in case of a reduced intraocular pressure. So these all mechanisms contribute to the transparency of the cornea. Okay, so how the corneal hydration is maintained? Okay, the cornea is in a state of relative dehydration. This relative dehydration is necessary for the corneal transparency. So normally the corneal water content, as we said, it is about 80%. This corneal transparency is kept, this water content in the cornea is kept at a balance by factors which draw water into the content, water into the cornea, like the swelling pressure and the intraocular pressure. You see the swelling pressure and the intraocular pressure try to push water into the cornea, and the factors which prevent the flow of water into the cornea, like the epithelial and the endothelial barrier. Along with it, there is an endothelial pump, that is the sodium potassium pump, which constantly pushes water, which draws water from the cornea and pushes into the aqueous. So these are all factors, that is the factors which try to push water into the cornea and the factors which try to remove water from the cornea, play a balance in order to maintain the transparency of the cornea. Now, coming to the stromal swelling pressure. The stromal swelling pressure is 50 millimeters of mercury, and it is exerted by the glycosaminoglycans and the collagen of the stroma. That is, the glycosaminoglycans are the matrix, extracellular matrix, and the collagens. They try to constantly repel each other because of their negatively charged molecule. They try to constantly repel each other and try to move away from each other. When they try to move away from each other, there is a swelling occurring in the cornea. This swelling causes movement of water into the cornea, and the imbibition pressure or the movement of water into the cornea is equal to the stromal pressure in vitro, but it is not the same in vivo. Okay. So next, evaporation of water from the corneal surface. There is a constant loss of loss of tear from the corneal surface, which increases its concentration of the precorneal tear fluid or the osmolarity. That is, the tear film becomes more that is more concentrated than the that is the water within the cornea. So this causes, that is, there is a more amount of salts in the tear film compared to the cornea. So this draws water from the cornea into the tear film. This constant drawing of water, water from the cornea is constantly drawn into the tear film. This constant drawing of water from the cornea into the tear film also maintains the transparency of the cornea. So the cornea is in a relative state of dehydration. Okay. So next, coming to the factors which lead to corneal edema. So what causes corneal edema? Okay. First is the intraocular pressure. When the intraocular pressure is more than the swelling pressure, there occurs corneal edema. When there is an increase in intraocular pressure, the water is pushed into the cornea, thereby causing edema of the cornea. The relation of swelling pressure of stroma to the intraocular pressure is the imbibition pressure. That is, the imbibition pressure of the corneal stroma is the intraocular pressure minus the swelling pressure. Okay. So as the stromal pressure decreases precipitously by increase in corneal thickness, mild corneal edema, which combined with increase in intraocular pressure leads to high imbibition pressures, and subsequently there is a formation of microbullae and epithelial edema. So water, when it is pushed into the cornea, it forms microbullae and epithelium becomes edematous. Okay. Apart from intraocular pressure, there are also cellular factors which affect the transparency. That is, the keratocytes. That is, the keratocytes, where are they located? They are located in between the lamellae. Okay, they also maintain the transparency by constantly producing collagens and proteoglycans. The keratocytes also contain enzymes which are involved in the assembly of the stromal matrix, and the specific enzyme defects are associated with corneal opacities. Example, in case of when there is a deficiency of polysaccharides, there occurs a corneal edema. Okay. Next, coming to corneal hydration. The corneal hydration is maintained, as you already said, by the barrier function of the epithelium and endothelium. This epithelium and endothelium are semi-permeable in nature, and they are all dependent on calcium for maintaining their barrier function. Okay. Apart from this, there are also active pump mechanisms. That is the mechanism by which the endothelium removes fluid from the stroma. This active process is by sodium potassium ATPase pump. The essential component of the endothelial pump function is the sodium potassium ATPase. This is an integral membrane protein. It is located in the basolateral aspect of the endothelium. Its action is vital in maintaining the corneal hydration. Central to all the system, as it maintains the sodium gradient required for the sodium hydrogen exchange, thereby promoting bicarbonate production. Apart from this, the second enzyme is the bicarbonate dependent ATPase. It is also essential for maintaining of the corneal thickness. The bicarbonate transported by the endothelium is generally intracellular via the action of carbonic anhydrase. This carbon dioxide diffuses into the cell from the extracellular space. It combines with the water in the presence of carbonic anhydrase. This carbonic anhydrase dissociates into the proton H plus ions and the bicarbonate ions. Apart from this, the third enzyme is a carbonic anhydrase enzyme. This inhibition of this enzyme causes decreased flow of fluid from the stroma to the aqueous. So when there is a inhibition of carbonic anhydrase enzymes, the fluid does not flow from the stroma to the aqueous, thereby causing corneal edema. Apart from the active process, There are also passive diffusion of potassium, chloride, and bicarbonate ions diffuse into the aqueous, and sodium, chloride, and bicarbonate ion diffuse from the aqueous into the cornea back. Okay. Now let us look at briefly about corneal pharmacology, like what happens when we apply topical drops on the cornea, and how are they metabolized, and how do they penetrate into the eye. Okay. Now the volume of a normal adult tear film is 7 to 9 microliters, and the maximum amount in the cul-de-sac that can be maintained is 25 to 30 microliters. So volume of a drop, a single drop is approximately 40 microliters. So most of the medication is immediately lost to the eyelashes, and they run to the eye, and the remainder is diluted by the tear film to approximately 25%. That is why we do not appreciate more than applying of more than one drop at a time. And for a medication to be effective, we ask it to be applied at regular intervals, but at one drop per time. Okay. So what happens is, because of the running away and because of wash away by the tear film, both the volume and concentration of the drops are reduced. Ointments are retained in a cul-de-sac to an extent. If you want a medication to be retained for a longer time, then it is more preferred as a petroleum jelly or an ointment based application. But though they are cumbersome, they are more sticky, and they cause a little bit of haziness over the cornea. Okay, so ointments are retained in the cul-de-sac, and they gradually melt, releasing the drug into the tears. The purpose is to increase the time the drug is present in the tears. After topical administration, most of the drug enters the stroma or the aqueous by corneal penetration. Drug penetrating to the conjunctiva is carried away by the blood vessels. The corneal epithelium provides the initial barriers due to its tight junction. Okay, the epithelium is composed of lipids, so non-polar substance penetrate readily. That is, those which do not split into the constituent ions. Okay, the stroma is contains mainly water. So for the stroma, the polar ions readily pass through. So drug should be easily present in both the non-polar, easily dissociable both in the non-polar and the polar form, so that it can be easily penetrating into the cornea. Okay, so drug must pass through both the barriers. Those soluble in lipids and water exhibit this best penetration. So it should be soluble both in drug should be soluble both in lipids and in water to penetrate the cornea easily. Okay, some preservatives present in the drugs like benzalkonium chloride, they impair the integrity of epithelial barrier, and they increase the penetration. So when you want a drug to penetrate, you have to destroy the epithelial barrier. Okay, so the penetration of a drug depends on the solubility of the drug. That is, the epithelium and endothelium are easily penetrated by lipid soluble substance, but the stroma, because it is hydrophilic, can allow only water soluble drug. So that hence the drug should be both amphipathic, that is, it should be both hydrophilic and hydrophobic, or it should be soluble both in lipids and in water. Okay, next is the ionization. A drug must have a capacity to exist in both the ionized and non-ionized form for better penetration. So non-ionized drugs can penetrate through epithelium, while the ionized drug only penetrate through the stroma. pH affects on the electrical charge and the stability of the solution. A solution outside the range of 4 to 10 increases the permeability. The size of the molecule, it is not important for lipid soluble substances, but for water soluble substances, they must be less than 4 angstroms. Molecular weight, a substance which is less than 500 milligrams can pass through less than 500 of molecular weight can pass through the cornea. Wetting agents. These wetting agents can also increase the permeability of the cornea by reducing the surface tension. Okay, so what happens with the aging in cases of cornea? With advancing age, the cornea becomes less translucent, and there is a dust-like opacification due to condensation of the stroma. There is a haziness happening in the cornea with aging. So both the Bowman's and the Decimate's membrane are also increased in their thickness. There is an arcus senilis, that is a wide ring-like arc present at the periphery of the cornea. This is due to the infiltration of extracellular lipids and it is universally present in all people above the age of 60 years. There is a small protrusion at the periphery of the Decimate membrane. These are known as Hazel-Henle bodies, and they do not interfere with the vision, unlike the central excursions which are present in Fuchs dystrophy. Okay. So that is all about the physiology of the cornea. Thank you for your attention from Dr. Wasim, Lakshminarayana Medical.